Yeah, as Michelle said, my name is Kate Tillotson Chavez. I'm an associate biologist here at Western Ecosystems. Um, and I put this presentation together with Rodney Richardson, who's a statistician and biologist at West, who is in the audience but is feeling under the weather. So I'm going to take the reins on this presentation today. And today we're talking about past, present, and the future of evaluating impacts of adaptive management on pollinators, with a focus on the Middle Rio Grande as well. And just making sure you all can see my next slide. I'm pretty sure you can. The challenge with pollinator conservation is that there are so many different species. And so trying to keep track of species, whether that be occurrence or range size, abundance, these are all questions that you might have when you're looking at a specific species. And then to scale that across, almost 5,000 species is really difficult. And so that is the main challenge when we're looking at pollinator conservation. But there are also a whole myriad of challenges, mainly focused on pollinator decline. There are lots of different impacts now that may impact pollinator decline. The main one is habitat loss, so conversion of land to maybe cropland. That is what this figure depicts. So those hotspots, red, orange, yellow, are where land is being converted to cropland, and then that is not necessarily viable habitat for pollinators. Climate change, which is having hard to predict impacts on species, and it's not the same across species. So climate change will be something that will really push the boundaries of our study with pollinator conservation. Parasites and pathogens. Parasites are often spread by farmed species of pollinators. And so this could be Apis mellifera or the honeybee, the European honeybee, or it could be even farmed bumblebee species that are used for pollination of crops. It's known that these species can then spread parasites and pathogens to native populations. And then also pesticides. So you can kind of see the linkage with habitat loss and conversion of habitat to cropland, and then the expanded use of pesticide on the landscape would really pose a threat to pollinators. So there are a couple of well-documented examples of decline, and then there are so many other more uncertain examples. The pictures here, we have the rusty patch bumblebee, the Carner blue butterfly, and then Taylor's checker spot. And these are all federally listed species where declines were tracked and monitored, and then they were proposed for listing and were listed under the Endangered Species Act. But for all these other uncertain species, listing is just hard across the board because pollinators are highly diverse groups. They're often highly speciose. Some of them are very small and obscure and can be very cryptic and hard to catch. They can also be hard to identify once you have caught them. So there's taxonomic expertise required when you are monitoring pollinators. And then some species, the adults, and maybe even also the larval forms, only forage on specific plant species. So if your pollinator, the pollinator that you're studying is maybe dependent on an annual plant species, then you're dependent on finding those plants, and then you can possibly monitor for those specific pollinators. There are a handful of federally imperiled pollinators in the Middle Rio Grande. The Sacramento Mountains checker spot is federally listed as endangered, and then the monarch butterfly is a candidate species, so it has yet to be listed, but is up for listing possibly this year in the fall. And then many different species of bumblebee have been petitioned for listing very recently. So you have the Morrison's southern variable cuckoo, American, and the western bumblebee. And then the large marble butterfly is also petitioned. And these have variable ranges throughout the Middle Rio Grande. For the state imperiled pollinators of the Middle Rio Grande and New Mexico, things get a lot murkier. New Mexico actually doesn't have any pollinators listed under their endangered and threatened species. However, there are species that occur within New Mexico and the Middle Rio Grande region that are imperiled or could be imperiled. It's just that data is possibly lacking. And a great example of this is the yucca giant skipper. This has a G5 ranking, which means that it is stable. And the G rankings are given by NatureServe, which documents species in lots of different taxonomic groups across the US. So it's got this globally stable ranking, but it's unranked in New Mexico, meaning that there's just not enough information. However, nearby states such as Nevada and Arizona have the yucca giant skipper ranked as imperiled and vulnerable to extinction or extirpation in those states. So more information is needed for the state of giant skipper populations within New Mexico. Similarly with the Anasazi crescent, which is given an imperiled ranking by NatureServe, but is unranked in New Mexico. Similarly with the bee species Osmia watsoni, which is only occurs in New Mexico and Texas, is unranked as far as state listing for both of those states, but is given an imperiled status by NatureServe. And so more information is also needed for that species. And then last but not least, as far as examples go, there's the obsolete viceroy butterfly, which is like not a very nice name to give a butterfly, but nonetheless, the taxonomic ranking is a 3. So that means that it is vulnerable to extinction or extirpation. And it has a state ranking in Nevada as 1, which is critically imperiled, so at high risk of extinction. But for New Mexico, a neighboring state, it has no ranking. And it is reliant on these riparian areas in these states because the host plant for the larval form of this species is salix or willow. So we have all these examples of species that need to be monitored and sampled for. And so how do we go about doing that? We could start with passive trapping methods. These are tried and true methods that have been widely used for a very long time. And there are many different options. We've got a couple examples here of pan traps. They have variable colors to attract different pollinators that may be attracted to different colors of flowers. There's a funnel trap there in the middle, and then there's a nesting trap. So this is generally for cavity nesting bees if you want to understand where they're nesting, maybe they'll understand their larval forms, or even what they are building their nests with. These are great traps to use. And what's great about a lot of these traps is that they cost very little to set up, and then there's generally a pretty high return. However, they do have some pitfalls. They are generally lethal, all of them, and their catch rate can be dependent on floral availability. So it's thought that if there are more flowers, you catch fewer bees. So those bees are visiting the floral resources available and not visiting your pan traps specifically, or maybe your funnel traps. And then these pan traps tend to be biased towards small bee species. So if you're monitoring for larger bees, you may not catch them. And these small bees are generally morphologically somewhat cryptic and hard to identify to species. But one of the biggest things is that if you're trying to understand endangered and imperiled species, it's not uncommon to kill many thousands of bees if you're employing these pan traps. And so that's a major pitfall for their use. Along with that, if you're collecting these massive amounts of specimens, then you need to identify them, and that can create a large taxonomic bottleneck as you wait for those identifications. So on the other side of this is active sampling, which is, I think, what everybody thinks of when they think of studying pollinators. Is you're out there with a net on a sunny day in a large stand of flowers. And you're not wrong. That really is what it looks like. But with active sampling, it often requires an experienced observer. So you need to be trained to look for pollinators, so that you're not just catching the very obvious ones. And unlike for this active sampling, it's not like your pan traps where you can just set it out, you can leave it, and then you can come back. You need to be out in the field. And when you are out in the field, every surveyor has their unique bias. And this could be almost as simple as maybe you're allergic to poison ivy, and so you're not sampling in a specific area, or you're avoiding a specific area. For these passive methods, generally there are less bees or pollinators sampled, and this leads to lower statistical power. And when you are trying to survey, you can only survey what's humanly possible. So maybe you don't have access to some trees. You may be restricted on the time of day. It's also effort. There's a restriction in effort. There can only be, you can only sample so much. So you may be restricted to certain times of the year, certain times of the day. You may only be able to sample during like good weather, things like that. So all of these are potential challenges when you're using active sampling. So we have active sampling, we have passive sampling, and then there is a new method of sampling that has grown in popularity over the last 5 to 10 years, and that is eDNA sampling, or environmental DNA sampling. And this was proposed largely for monitoring in aquatic environments, but has spread to terrestrial environments, and has spread to the pollinator world, and where you can take generally flowers or other plant material and sample that for pollinators, because this environmental DNA is sampling DNA that's been left behind. It's sort of like a crime scene, there will be human DNA left behind at a crime scene. It's the same for pollinator visiting a flower. They're going to leave behind possibly feces or scales. So for your Lepidoptera, um, all of that DNA gets left behind and can be sampled for successfully. And so for Rodney's research, he used a paired sampling method where they did active sampling with nets, and then they were also sampling for pollinators with eDNA. And so they were really comparing and contrasting these two methods, and if they were successful in detecting pollinators, but also specifically Bombus affinis, so the rusty patch bumblebee. And they were doing this in Central Appalachia. So Central Appalachia is one of the last areas with known occurrence of Bombus affinis in the east. And you can see here that with those active sampling methods, they've collected some Bombus specimens. They're still alive, and they can go ahead and look at those species. They can look at them and evaluate, possible pathogen load. They can look at the sex of the specimen, and they can even swab the pollen loads to see which flowers the bee is visiting. And so for these for this eDNA, they found that when they were using these specific primers, so these DNA sequences that were bee specific, they had 286 occurrences or detections of bees, and they were able to link these. And here they're sorted into families with the flower families that they were visiting. And so these were samples of flowers that were taken from the field and then processed for bee presence using these bee-specific DNA primers. But what they also found was that 28S bee-specific primer also picked up or detected Lepidoptera as well. And so there was a little bit less resolution, but it was very good at detecting Lepidoptera, and they collected, a pretty diverse array of Lepidoptera across families as they were sampling for these bees. And in using a bumblebee-specific COI primer, so another DNA primer, they were able to compare whether they were getting the same detection rates with netting as they were with eDNA. And while there are some differences with Bombus impatiens, for Bombus affinis, you can see that the detection rate was very similar, which means that it was pretty effective as a method for understanding occurrence of Bombus affinis on the landscape. And it was just as practical a method as using the more traditional active netting sampling. So there are benefits and challenges when using eDNA, just like any other surveying method. And some of the benefits you have that this is really ideal for sensitive ecosystems where you may be sampling for threatened and endangered species. So you maybe do not want to take those species off the landscape, and it's very low impact. And so that makes it great for sampling in these environments and for those specific species. it is also great for data-hungry models. So for occupancy models and things like that, you can feed those models a lot of data because you're taking, because you're just able to take so much more data with this eDNA and collect those occurrences across the landscape. Some challenges, though, are that there's only a limited set of study systems as of now where this has been demonstrated beyond a proof of concept. So it hasn't been repeated enough to maybe say that this is a tried and true method for pollinator surveys specifically. It also may be cost prohibitive for some organizations. It's still somewhat expensive to use on a large scale. And it also doesn't capture reference organisms for future study. So this could be for morphological study. It could be to understand pollen loads or pathogen loads. All of these things help us understand the biology of these different pollinators. However, it has grown in popularity over the last 10 years, and it's something that I'm excited to see used more in the future. So Rodney wanted to acknowledge his collaborators in some of this work and also his funding. And we'd just like to thank you for allowing us to present our work.